Welcome to our Class 12 Biology Revision Series. Hello students. Welcome to the channel. If you're preparing for the CBSE Class 12 Biology Board exam, you've come to the right place. This series is specially designed for students from remote and rural areas, where quality study resources may not always be easily available. Here, we explain every important concept in a simple, clear, and exam-focused way. Our goal is to help you revise smarter, improve your confidence, and score your best in the board exams. So, keep your notebook ready, stay focused, and let's begin today's revision. Detailed Summary. Unit V, Reproduction and Chapter 1. Sexual Reproduction in Flowering Plants, Unit Overview and Biographical Highlight. Unit V. Reproduction, General Overview. Reproduction is a vital biological process that ensures the continuation of species over millions of years despite the death of individual organisms. It occurs through asexual or sexual modes, with sexual reproduction offering the critical advantage of creating genetic variations that enhance survival. The unit focuses on reproductive mechanisms in flowering plants and humans, along with reproductive health. Biography. Panchanan Maheshwari, 1904–1966, background. Born in Jaipur, Rajasthan, he became one of the world's most distinguished botanists. He earned his DSc from Allahabad University under the mentorship of Dr. W. Dudgeon. Key scientific contributions: popularized the use of embryological characters in plant taxonomy. Established the Department of Botany at the University of Delhi as a world-renowned center for research in plant embryology and tissue culture. Achieved worldwide acclaim for pioneering work on test tube fertilization and intra-ovarian pollination. Legacy. Elected Fellow of the Royal Society of London, FRS, and led the publication of the first NCERT biology textbooks for higher secondary schools in 1964. Chapter 1. Sexual Reproduction in Flowering Plants. 1. Flower, a fascinating organ of angiosperms. Flowers are sites of sexual reproduction that evolved to facilitate the formation of seeds and fruits. Structurally, flowers consist of male reproductive organs, androecium, and female reproductive organs, gynoecium. 2. Pre-fertilization. Structures and events. A. Stamen, microsporangium, and pollen grain. Stamen structure consists of a slender stalk called the filament and a terminal, usually bilobed structure called the anther. Anther architecture. A typical angiosperm anther is bilobed and the cous, 2 theca per lobe, making it a 4-sided, tetragonal, structure containing 4 microsporangia, 2 in each lobe. Microsporangia develop into pollen sacs. Microsporangium wall layers. 1. Epidermis, endothecium, and middle layers. Outer 3 layers that protect the structure and assist in anther dehiscence to release pollen. 2. Tapetum. Innermost layer with dense cytoplasm and multiple nuclei that provides nourishment to developing pollen grains. Microsporogenesis. Young microsporangia contain central sporogenous tissue. Cells of this tissue, pollen mother cells or PMCs, undergo meiosis to form haploid microspore tetrads. As the anther matures and dehydrates, microspores separate to become pollen grains. Pollen grain, male gametophyte, size and exene, spherical, 25 to 50 micrometers in diameter. The outer layer, exene, is composed of sporopollenin, an extremely durable organic material resistant to high temperatures, strong acids, alkalis, and enzymatic degradation. The exene contains openings called germ pores where sporopollenin is absent. In time, thin, continuous inner wall composed of cellulose and pectin. Cellular composition. Mature pollen contains a large vegetative cell, rich in food reserves, and a smaller, floating spindle-shaped generative cell. About 60% of angiosperms shed pollen at this 2-celled stage, while others divide mitotically to shed at a 3-celled stage, containing 2 male gametes. Relevance and applications. Can cause respiratory allergies and conditions like asthma, e.g., caused by invasive parthenium or carrot grass. Rich in nutrients, used commercially as performance-enhancing supplements for athletes and racehorses. Pollen viability ranges from 30 minutes, rice, wheat, to several months, Rosacea, Leguminosae, Solanaceae. Pollen can be preserved for years in liquid nitrogen, 196 carat, CIRCC, in pollen banks. B. The pistil, megasporangium, ovule, and embryo sac. Gynoecium and pistil represents the female reproductive organ. It can be monocarpellary, single pistil, or multicarpellary, multiple pistils, which may be fused as syncarpous or free as apicarpus. Pistil components. Stigma, terminal landing platform for pollen. Style, elongated neck connecting stigma to ovary. Ovary, basal swollen portion containing the ovarian cavity, locule, and placenta. Structure of the ovule, megasporangium, attached to the placenta via a stalk called the funicle. The junction between the ovule body and funicle is the hilum. Protected by 1 or 2 integuments, which encircle the central nucellus except at a small apical opening called the micropyle. The basal end opposite the micropyle is the chalaza. Megasporogenesis and female gametophyte development. A single megaspore mother cell, MMC, in the micropylar nucellus undergoes meiosis to produce 4 haploid megaspores. In most species, 3 megaspores degenerate, and only 1 functional megaspore undergoes monosporic development to form the female gametophyte, embryo sac. The functional megaspore nucleus undergoes 3 free nuclear meiotic divisions forming an 8-nucleate structure. Mature embryo sac structure, 7-celled, 8-nucleate. Egg apparatus, micropylar end, consists of 2 synergids, containing a filiform apparatus to guide pollen tube entry, and 1 egg cell. Antipodals, chalazole end, 3 cells located at the base. Central cell. Large central cell containing 2 polar nuclei. See pollination mechanisms and outbreeding. Definition. Transfer of pollen grains from anther to stigma. Types of pollination. 1. Autogamy. Self-pollination within the same flower. Occurs in open, chasmogamous, flowers or non-opening, cleistogamous, flowers that guarantee seed production without pollinators. 2. Gytonogamy. Pollen transfer to another flower on the same plant, functionally cross-pollination, genetically autogamy. 3. Xenogamy. Pollen transfer to a flower on a different plant, bringing genetically distinct pollen. Pollination agents. Abiotic. Wind. Pollen is light and non-sticky, stamens well exposed, stigmas feathery, and water. Rare. Seen in plants like Vallisneria, Hydrilla, and marine seagrasses like Zostera. Biotic. Animals and insects. Flowers are large, colorful, fragrant, and produce nectar. Outbreeding devices. Mechanisms evolved to prevent continuous self-pollination and inbreeding depression. These include non-synchronized pollen release and stigma receptivity, differing spatial positions of anthers, stigmas, genetic self-incompatibility, and dioecious, unisexual plants. D. Pollen pistil interaction and artificial hybridization. Pollen pistil interaction. Dynamic chemical recognition process where the stigma determines pollen compatibility, promoting germination for compatible pollen or inhibiting incompatible pollen. Pollen tube growth. Compatible pollen germinates via germ pore. The pollen tube grows through styled tissues into the ovary and enters a synergid via the filiform apparatus. Artificial hybridization. Plant breeding technique involving emasculation, removal of anthers from female bisexual flower buds before dehiscence. Bagging. Covering emasculated or female flowers with paper bags to prevent unwanted pollination before dusting desired pollen onto receptive stigmas. 3. Double fertilization. Double fertilization is an event unique to flowering plants that involves two distinct fusion events. 1. Syngamy. One male gamete fuses with the egg cell nucleus to produce a diploid zygote. 2. Triple fusion. The second male gamete fuses with the two polar nuclei in the central cell to form a triploid primary endosperm nucleus, pen. Following double fertilization, the central cell becomes the primary endosperm cell, PEC, which develops into endosperm, while the zygote develops into an embryo. 4. Post-fertilization. Structures and events. A. Endosperm development. Endosperm development precedes embryo development to ensure a steady supply of nutrients. Types. Successive free nuclear divisions yield free nuclear endosperm, e.g., coconut water, which later undergoes cell wall formation to become cellular endosperm, e.g., coconut kernel. Endosperm may be completely consumed before seed maturity, pea, groundnut, beans, or persist in mature seeds, castor, coconut. B. Embryo development occurs at the micropylar end of the embryo sac through stages: preembryo to globular to heart-shaped to mature embryo. Dicotyledonous embryo structure consists of an embryonal axis and two cotyledons. The region above the cotyledons is the epicotyl, terminates at the plumule, and the region below is the hypocotyl, terminates at the radical, root cap. Monocotyledonous embryo structure possesses a single cotyledon called the scutellum situated laterally. The lower embryonal axis, radical and root cap, is enclosed in a protective sheath called coleorhiza, while the upper shoot tip, epicotyl, is enclosed in coleoptile. Chapter 2 here is a detailed, section-by-section summary of the provided text on human reproduction. Overview of human reproduction. Humans are sexually reproducing and viviparous organisms. The human reproductive process follows a defined sequence of events. 1. Gametogenesis. Formation of male sperms and female ova. 2. Insemination. Transfer of sperms into the female genital tract. 3. Fertilization. Fusion of male and female gametes to form a zygote. 4. Implantation. Formation and attachment of the blastocyst to the uterine wall. 5. Gestation. Embryonic development inside the mother's body. 6. Parturition. Delivery of the baby. These events initiate after puberty. While sperm formation continues into old age in men, ovum formation ceases in women around the age of 50 years. 1. The male reproductive system. The male reproductive system is located in the pelvic region and consists of the testes, accessory ducts, accessory glands, and external genitalia. Testes and scrotum. The primary sex organs, paired testes, lie outside the abdominal cavity within a pouch called the scrotum. The scrotum maintains the testes at a temperature 2 to 2.5 degrees Celsius lower than body temperature, which is required for spermatogenesis. Adult testes are oval, 4 to 5 centimeters long, 2 to 3 centimeters wide, and enclosed by a dense covering. Each testis contains about 250 testicular lobules, each housing 1 to 3 highly coiled seminiferous tubules. Cellular structure of seminiferous tubules. Spermatogonia, male germ cells, undergo meiotic divisions to produce sperms. Sertoli cells provide nutrition to developing germ cells. Leydig cells, interstitial cells, located in spaces outside the tubules, synthesize and secrete male sex hormones, androgens. Immunologically competent cells are also present here. Male accessory ducts and glands. Duct pathway. Seminiferous tubules to rete testis to vasa efferentia to epididymis to vas deferens to ejaculatory duct to urethra, terminating at the urethral meatus. Accessory glands. Paired seminal vesicles, a single prostate gland, and paired bulbourethral glands. Seminal plasma. Their combined secretion forms seminal plasma, which is rich in fructose, calcium, and specific enzymes. Bulbourethral secretions also aid in penis lubrication. External genitalia. The penis is made of erectile tissue that facilitates erection for insemination. Its enlarged tip is called the glans penis, covered by a loose skin fold called the foreskin. 2. The female reproductive system located in the pelvic region. This system includes the primary sex organs, ovaries, accessory ducts, external genitalia, and integrated mammary glands. Ovaries. Primary female sex organs that produce ova and steroid ovarian hormones. Located on each side of the lower abdomen, measuring 2 to 4 centimeters in length, and attached to the pelvic wall and uterus by ligaments. Covered by thin epithelium enclosing the ovarian stroma, which is divided into an outer peripheral cortex and an inner medulla. Female accessory ducts. Fallopian tubes, oviducts, extend from the ovaries to the uterus. Divided into the fimbriae, finger-like projections to collect the ovum, infundibulum, ampulla, wider middle region, and isthmus, narrow region joining the uterus. Uterus, womb, an inverted pear-shaped single organ supported by pelvic ligaments. Its wall consists of 3 layers. Perimetrium, outer thin membranous layer. Myometrium, middle thick smooth muscle layer, exhibits strong contractions during delivery. Endometrium, inner glandular layer, undergoes cyclical changes during menstruation. Cervix and vagina. The cervical cavity, cervical canal, together with the vagina forms the birth canal. Female external genitalia, vulva, includes the mons pubis, fatty tissue covered by pubic hair, labia majora, fleshy tissue folds, labia minora, inner paired folds, hymen, membrane partially covering vaginal opening, and clitoris, tiny finger-like structure above the urethral opening. Note: the hymen can break during physical activities, intercourse, or tampon insertion, making its presence or absence an unreliable indicator of virginity. Mammary glands. Functional paired breasts containing glandular tissue and variable fat. Divided into 15 to 20 mammary lobes containing clusters of milk-secreting cells called alveoli. Pathway. Alveoli to mammary tubules to mammary duct to mammary ampulla to lactiferous duct to nipple. 3. Gametogenesis. Gametogenesis is the process of gamete formation, spermatogenesis in males and oogenesis in females. Spermatogenesis begins at puberty due to a rise in gonadotropin-releasing hormone, GnRH, from the hypothalamus. GnRH stimulates the anterior pituitary to secrete LH, acts on Leydig cells to release androgens, and FSH, acts on Sertoli cells to stimulate spermiogenesis factors. Developmental steps. 1. Spermatogonia, 2n equals 46, multiply by mitosis. 2. Primary spermatocytes, 2n equals 46, undergo first meiotic division to form 2 equal haploid secondary spermatocytes, n equals 23. 3. Secondary spermatocytes undergo second meiotic division to form 4 equal haploid spermatids, n equals 23. 4. Spermiogenesis. Transformation of spermatids into functional spermatozoa, sperms. 5. Spermiation. Sperm heads embed in Sertoli cells and are released from seminiferous tubules. Structure of a sperm. Composed of a head, contains haploid nucleus and an acrosome filled with fertilization enzymes. Neck. Middle piece, packed with mitochondria for energy, motility, and tail. A normal ejaculation contains 200 to 300 million sperms. Normal fertility requires at least 60% normal shape, size and 40% vigorous motility. Oogenesis, initiated during embryonic development. A few million gamete mother cells, oogonia, form in fetal ovaries. No oogonia are formed after birth. Oogonia enter meiotic prophase I and get arrested as primary oocytes. Follicular maturation. 1. Primary follicle. Primary oocyte surrounded by a layer of granulosa cells. Most degenerate before puberty, leaving 60,000 to 80,000 per ovary. 2. Secondary follicle. Primary follicle surrounded by more granulosa cells and a new theca. 3. Tertiary follicle. Characterized by a fluid-filled cavity, antrum, and inner-outer theca layers. The primary oocyte completes its first meiotic division here, forming a large haploid secondary oocyte and a small first polar body. 4. Graphian follicle. Mature follicle containing the secondary oocyte surrounded by a zona pellucida layer. Ovulation. The Graphian follicle ruptures to release the secondary oocyte, ovum, into the pelvic cavity. 4. The menstrual cycle. The regular reproductive cycle in female primates averages 28 to 29 days. The first cycle is called menarche, at puberty, and cycle cessation around age 50 is menopause. Here is the breakdown of the menstrual cycle phases described in full text. The 4 phases of the menstrual cycle. The human menstrual cycle averages 28 to 29 days and progresses through 4 main phases. 1. Menstrual phase. Days 1 to 5. This phase marks the beginning of the cycle and usually lasts from day 1 to day 5. It is characterized by the breakdown and shedding of the inner uterine lining, the endometrium, along with its blood vessels, resulting in menstrual flow through the vagina. This phase occurs when the ovum released in the previous cycle remains unfertilized. 2. Follicular or proliferative phase, days 6 to 13. Lasting from day 6 to day 13. This phase involves 2 simultaneous processes. In the ovaries, primary follicles mature into fully developed Graphian follicles under the influence of pituitary hormones, specifically FSH, follicle-stimulating hormone, and LH, luteinizing hormone. In the uterus, the endometrium regenerates and thickens through cellular proliferation. As the follicles grow, they secrete increasing amounts of estrogen. 3. Ovulatory phase, day 14. Occurring at the midpoint of the cycle around day 14, both FSH and LH reach peak levels in the body. A rapid increase in LH, referred to as the LH surge, triggers the rupture of the matured Graphian follicle, releasing the secondary oocyte, ovum, into the pelvic cavity. This event is known as ovulation. 4. Luteal or secretory phase, days 15 to 28. Spanning from day 15 to day 28, the remnants of the ruptured Graphian follicle transform into a temporary endocrine organ called the corpus luteum. The corpus luteum secretes high amounts of progesterone, which maintains a thick, vascular endometrium necessary for embryo implantation and pregnancy. If fertilization does not occur, the corpus luteum degenerates, leading to a drop in progesterone levels. This breakdown triggers the start of the next menstrual phase, restarting the cycle. If fertilization does not occur, the corpus luteum degenerates, causing endometrial disintegration and initiating a new cycle. Menstruation stops during pregnancy. 5. Fertilization and implantation, fertilization site. Semen deposited in the vagina travels through the cervix and uterus to the ampullary region of the fallopian tube, where fertilization occurs if sperm and ovum arrive simultaneously. Sperm entry. Sperm contacts the zona pellucida, triggering membrane changes that block additional sperms, prevents polyspermy. Acrosomal secretions allow the sperm nucleus to enter the secondary oocyte cytoplasm. This induces completion of the second meiotic division of the secondary oocyte, releasing a second polar body and a haploid otid, ovum. The sperm and ovum nuclei fuse to form a diploid zygote, 2n equals 46. Sex determination. Females produce X-bearing ova. Males produce 50% X-bearing sperms and 50% Y-bearing sperms. Fusion of X sperm gives an XX female. Y sperm gives an XY male. Thus, the genetic sex is determined entirely by the father. Cleavage to implantation. As the zygote moves through the isthmus toward the uterus, meiotic divisions, cleavage, produce daughter cells called blastomeres, 2, 4, 8, 16 cells. A solid embryo with 8 to 16 blastomeres is a morula. The morula develops into a blastocyst, featuring an outer trophoblast layer and an inner cell mass. The trophoblast attaches to the endometrium, and rapid uterine cell growth embeds the blastocyst into the uterine wall, implantation, initiating pregnancy. 6. Pregnancy and embryonic development, placenta formation. Trophoblast forms finger-like chorionic villi, which interdigitate with maternal uterine tissues to create the placenta. Connected to the fetus via the umbilical cord. Functions. Facilitates O_2 and nutrient delivery to the fetus and waste removal, CO_2, excretory waste. Endocrine functions of placenta. Secretes human chorionic gonadotropin, HCG, human placental lactogen, HPL, estrogens, and progestogens. The ovary secretes relaxin during late pregnancy. Note: HCG, HPL, and relaxin are produced only during pregnancy. Maternal levels of cortisol, prolactin, thyroxine, estrogen, and progesterone also rise significantly. Germ layers and stem cells. Inner cell mass differentiates into 3 primary germ layers: ectoderm, outer, endoderm, inner, and mesoderm, middle, which give rise to all adult tissues. Contains potent stem cells. Fetal development timeline, 9 months, month 1. Heart is formed, heartbeats detectable via stethoscope. Month 2. Limbs and digits develop. Month 3. End of first trimester, 12 weeks. Major organ systems form, limbs, external genitalia. Month 5. First fetal movements and head hair appear. Month 6. End of second trimester, 24 weeks. Fine body hair grows, eyelids separate, and eyelashes form. Month 9. Fetus is fully developed and ready for birth. 7. Parturition and lactation. Parturition, childbirth, induced by a complex neuroendocrine mechanism. Signals from the mature fetus and placenta induce mild uterine contractions called the fetal ejection reflex. This triggers oxytocin release from the mother's posterior pituitary. Oxytocin stimulates stronger uterine contractions, promoting further oxytocin release, a positive feedback loop that expels the baby through the birth canal, followed by the placenta. Lactation. Mammary glands differentiate during pregnancy and begin milk production near its end. Colostrum. The initial milk produced during early days of lactation. It is rich in antibodies crucial for providing passive immunity and disease resistance to the newborn. Chapter 3. Here is a comprehensive summary and key takeaway guide for Chapter 3. Reproductive health from the provided textbook material. Overview of Chapter 3. Reproductive Health 1. Reproductive health. Problems and strategies. According to the World Health Organization, WHO, reproductive health refers to total well-being in all aspects of reproduction, including physical, emotional, behavioral, and social aspects. India was among the first countries to initiate national action plans for total reproductive health, starting with the Family Planning Program in 1951. These initiatives are currently operated under the Reproductive and Child Healthcare (RCH) programs. Amniocentesis is a procedure used to analyze fetal cells and amniotic fluid to detect genetic disorders such as Down syndrome, sickle cell anemia, and hemophilia. A statutory ban on amniocentesis for sex determination was legally enacted to check female feticides. Saheli, a non-steroidal oral contraceptive pill taken once a week, was developed by scientists at the Central Drug Research Institute, CDRI, in Lucknow, India. 2. Population stabilization and birth control, rapid declines in death rate, maternal mortality rate, MMR, and infant mortality rate, IMR, along with an increase in the number of people of reproducible age, contributed to population growth. Ideal contraceptives should be user-friendly, easily available, effective, reversible with minimal side effects, and non-interfering with sexual drive. Contraceptive categories: natural, traditional methods, periodic abstinence, avoiding coitus from day 10 to 17 of the menstrual cycle, the fertile period, withdrawal, coitus interruptus, male partner withdraws the penis before ejaculation, lactational amenorrhea, absence of menstruation during intense lactation, effective up to 6 months post-parturition. Barrier methods: condoms, thin rubber, latex sheaths covering the penis or vagina, cervix. Also protect users from STIs and AIDS, e.g., neurode. Diaphragms, cervical caps, and vaults. Reusable rubber barriers inserted into the female reproductive tract to cover the cervix. Intrauterine devices, IUDs. Non-medicated IUDs, e.g., Lippes loop. Copper-releasing IUDs, e.g., CUT, Cu7, Multilode 375. Released copper ions suppress sperm motility and fertilizing capacity. Hormone-releasing IUDs, e.g., Progestazert, LN20. Make the uterus unsuitable for implantation and cervix hostile to sperms. Oral contraceptives. Small doses of progestogens or progestogen-estrogen combinations taken daily for 21 days. Injectables and implants. Subcutaneous implants or injections of progestogens with longer effective periods. Emergency contraceptives. Administration of progestogens, progestogen-estrogen combinations, or IUDs within 72 hours of coitus. Surgical methods: sterilization, vasectomy. Removal or tying up of a small part of the vas deferens in males. Tubectomy. Removal or tying up of a small part of the fallopian tubes in females. 3. Medical termination of pregnancy, MTP. Intentional or voluntary termination of pregnancy before full term is called MTP or induced abortion. Government of India legalized MTP in 1971 with strict conditions to prevent its misuse, such as female feticide. MTPs are considered relatively safe during the first trimester, up to 12 weeks of pregnancy. Under the MTP Amendment Act, 2017, termination requires the opinion of one registered medical practitioner if within 12 weeks, or 2 practitioners if between 12 and 24 weeks. 4. Sexually transmitted infections, STIs. Diseases transmitted through sexual intercourse are called STIs. Venereal diseases, VD, or reproductive tract infections, RTI. Common STIs include gonorrhea, syphilis, genital herpes, chlamydiasis, genital warts, trichomoniasis, hepatitis B, and HIV. Hepatitis B, genital herpes, and HIV infections are not completely curable. High incidence of STIs is reported among individuals in the age group of 15 to 24 years. Untreated STIs can lead to pelvic inflammatory diseases, PID, ectopic pregnancies, stillbirths, infertility, or reproductive tract cancer. 5. Infertility and assisted reproductive technologies, ART. Infertility is defined as the inability to produce children despite unprotected sexual cohabitation. Special ART techniques: in vitro fertilization, IVF, and embryo transfer, ET. ZIFT, zygote intrafallopian transfer, transfer of zygote or early embryo, up to 8 blastomeres, into the fallopian tube. IUT, intrauterine transfer, transfer of embryos with more than 8 blastomeres into the uterus. GIFT, gamete intrafallopian transfer, transfer of an ovum collected from a donor into the fallopian tube of another female. ICSI, intracytoplasmic sperm injection, direct injection of a sperm into the ovum in the laboratory. Artificial insemination, AI, IUI, Intrauterine insemination, semen collected from the husband or donor is artificially introduced into the vagina or uterus. Chapter 4. Summary. Principles of Inheritance and Variation. This text-based guide details the fundamental concepts of genetics, including how traits are passed between generations and the factors contributing to genetic variation. 1. Mendel's laws of inheritance. Gregor Mendel conducted hybridization experiments using the garden pea plant, Pisum sativum, between 1856 and 1863. By analyzing large sample sizes with statistical logic, he established the basic rules of biological inheritance. Mendel focused on 14 true breeding pea varieties across 7 contrasting pairs of traits: stem height, tall versus dwarf, flower color, violet versus white, flower position, axial versus terminal, pod shape, inflated, full, versus constricted, pod color, green versus yellow, seed shape, round versus wrinkled, seed color, yellow versus green. Monohybrid cross concepts. A monohybrid cross examines a single trait pair, such as tall times dwarf, F_1 generation, filial 1, progeny display only one parental trait, all tall, without blending. F_2 generation, filial 2, self-pollinating F_1 offspring allows the hidden recessive trait to reappear, yielding a 3:1 phenotypic ratio. 3 tall, 1 dwarf, and a 2:1 genotypic ratio. 1/4 TT, 1/2 TT, 1/4 TT. Factors and alleles. Mendel proposed that physical units called factors, now known as genes, pass unchanged through gametes. Alternative versions of a gene are called alleles. Test cross. Performed by crossing an individual showing a dominant trait with a homozygous recessive parent to verify if its genotype is homozygous, TT, or heterozygous, TT. The 3 Mendelian laws. 1. Law of dominance. Traits are governed by discrete factors that occur in pairs. When 2 contrasting factors are present, heterozygous state, one allele masks the expression of the other. 2. Law of segregation. Alleles do not blend. During gamete formation, meiosis, the 2 alleles segregate so that each gamete receives only one allele. 3. Law of independent assortment. Derived from dihybrid crosses, e.g., round yellow times wrinkled green. It states that the segregation of one pair of traits occurs independently of any other pair, resulting in an F_2 phenotypic ratio of 9:3, 3:1. 2. Deviations from Mendelian inheritance, incomplete dominance. The F_1 phenotype is an intermediate blend between both parents. For example, in snapdragons, Antirrhinum sp. crossing red flowers, RR, with white flowers, RR, yields pink flowers, RR. The F_2 phenotypic and genotypic ratios are identical, 1:2:1. County dominance. Both alleles in a heterozygous pair express themselves fully at the same time. An example is human ABO blood grouping, where alleles I-A and I-B co-express to produce the AB blood type. Multiple alleles. Occurrence of 3 or more alleles controlling the same gene across a population. Human ABO blood types are governed by 3 alleles, I-A, I-B, and I, giving rise to 6 genotypes and 4 phenotypes. Pleiotropy. A single gene influences multiple, seemingly unrelated phenotypic traits by affecting underlying metabolic pathways. For instance, phenylketonuria, PKU, is caused by a single gene mutation that leads to mental retardation as well as reduced hair and skin pigmentation. Polygenic inheritance. Trait variation controlled by 3 or more genes with cumulative additive effects, often impacted by environmental factors. Examples include human skin color, influenced by genes A, B, C, and human height. 3. Chromosomal theory, linkage and recombination, rediscovery, 1900. Hugo de Vries, Karl Correns, and Erich von Tschermak independently rediscovered Mendel's principles. Chromosomal theory of inheritance, formulated by Walter Sutton and Theodore Boveri, 1902, who observed that the behavior of chromosomes during cell division mirrors the segregation patterns of Mendelian genes. Experimental proof. Thomas Hunt Morgan confirmed the theory using fruit flies, Drosophila melanogaster, due to their short 2-week life cycle, high reproduction rate, clear sexual dimorphism, and distinct hereditary traits. Linkage. The physical association of genes on the same chromosome that prevents them from assorting independently. Recombination. The process by which non-parental allele combinations are generated through meiosis. Genetic mapping. Alfred Sturtevant used recombination frequencies between linked genes to measure physical distances and create gene maps, a foundation for modern genome sequencing. 4. Mechanisms of sex determination. Types of systems. Male heterogamety. Males produce 2 distinct types of gametes. OXY system. Females are XX and males are XY, e.g., humans and Drosophila. OXO system. Females are XX and males have a single X chromosome designated as XO, e.g., grasshoppers. Female heterogamety, ZW system. Females produce 2 distinct types of gametes, ZW, while males carry 2 matching sex chromosomes, ZZ, e.g., birds. Haplodiploid system. Observed in honeybees. Females are diploid, 2n equals 32, and hatch from fertilized eggs. Males, drones, are haploid, n equals 16, and develop from unfertilized eggs via parthenogenesis. Because drones produce sperm through mitosis, they lack fathers and sons, but have grandfathers and grandsons. Sex determination in humans. Humans possess 22 pairs of autosomes and 1 pair of sex chromosomes. Females carry 2 X chromosomes, XX, while males carry 1 X and 1 Y chromosome, XY. Every pregnancy carries an equal 50% probability of conceiving a male or female, dictated solely by whether an X or Y-bearing sperm fertilizes the egg. 5. Mutations and pedigree analysis. Mutation. An alteration in the DNA sequence that leads to changes in genotype and phenotype. Mutations can be point mutations, alteration of a single base pair, or frame shift mutations, insertions or deletions of bases. Mutagens, such as UV radiation, induce these DNA changes. Pedigree analysis. A tree diagram mapping inheritance patterns of specific traits or genetic conditions across generations of a family line. 6. Human genetic disorders. A. Mendelian disorders, single gene defects. Color blindness. An X-linked recessive disorder impairing red or green cone photoreceptors. Affects roughly 8% of males and 0.4% of females. Hemophilia. An X-linked recessive condition that impairs essential blood clotting factors, causing prolonged bleeding from minor injuries. Sickle cell anemia. An autosomal recessive condition caused by a point mutation swapping glutamic acid, GLU, for valine, VAL, at the 6th position of the beta-globin chain, GAG to GUG. Red blood cells deform into a sickle shape under low oxygen levels. Phenylketonuria, PKU, an autosomal recessive metabolic disorder caused by the absence of the enzyme phenylalanine hydroxylase, resulting in phenylalanine buildup and severe mental retardation. Thalassemia. An autosomal recessive quantitative blood disorder marked by reduced synthesis of alpha or beta globin chains, leading to severe anemia. B chromosomal disorders, abnormal chromosome counts, caused by non-disjunction during cell division, resulting in the addition, trisomy, or loss, monosomy, of whole chromosomes. Down syndrome, trisomy 21, caused by an extra copy of chromosome 21, total of 47 chromosomes. Traits include short stature, a small rounded head, furrowed tongue, broad palm crease, and physical, mental developmental delays. Klinefelter's syndrome, 47, XE, caused by an extra X chromosome in males. Traits include an overall masculine body with development of feminine characteristics, e.g., gynecomastia, individuals are sterile. Turner's syndrome, 45, XO, caused by the absence of one X chromosome in females. Traits include sterility due to underdeveloped, rudimentary ovaries, and a lack of secondary sexual characteristics. Chapter 5. Summary. Principles of Inheritance and Variation. This text-based guide details the fundamental concepts of genetics, including how traits are passed between generations and the factors contributing to genetic variation. 1. Mendel's laws of inheritance. Gregor Mendel conducted hybridization experiments using the garden pea plant, Pisum sativum, between 1856 and 1863. By analyzing large sample sizes with statistical logic, he established the basic rules of biological inheritance. Mendel focused on 14 true breeding pea varieties across 7 contrasting pairs of traits: stem height, tall versus dwarf, flower color, violet versus white, flower position, axial versus terminal, pod shape, inflated, full, versus constricted, pod color, green versus yellow, seed shape, round versus wrinkled, seed color, yellow versus green. Monohybrid cross concepts. A monohybrid cross examines a single trait pair, such as tall times dwarf, F_1 generation, filial 1, progeny display only one parental trait, all tall, without blending. F_2 generation, filial 2, self-pollinating F_1 offspring allows the hidden recessive trait to reappear, yielding a 3:1 phenotypic ratio. 3 tall, 1 dwarf, and a 2:1 genotypic ratio. 1/4 TT, 1/2 TT, 1/4 TT. Factors and alleles. Mendel proposed that physical units called factors, now known as genes, pass unchanged through gametes. Alternative versions of a gene are called alleles. Test cross. Performed by crossing an individual showing the dominant trait with a homozygous recessive parent to verify if its genotype is homozygous, TT, or heterozygous, TT. The 3 Mendelian laws. 1. Law of dominance. Traits are governed by discrete factors that occur in pairs. When 2 contrasting factors are present, heterozygous state, one allele masks the expression of the other. 2. Law of segregation. Alleles do not blend. During gamete formation, meiosis, the 2 alleles segregate so that each gamete receives only one allele. 3. Law of independent assortment. Derived from dihybrid crosses, e.g., round yellow times wrinkled green. It states that the segregation of one pair of traits occurs independently of any other pair, resulting in an F_2 phenotypic ratio of 9:3, 3:1. 2. Deviations from Mendelian inheritance, incomplete dominance. The F_1 phenotype is an intermediate blend between both parents. For example, in snapdragons, Antirrhinum sp. crossing red flowers, RR, with white flowers, RR, yields pink flowers, RR. The F_2 phenotypic and genotypic ratios are identical, 1:2:1. County dominance. Both alleles in a heterozygous pair express themselves fully at the same time. An example is human ABO blood grouping, where alleles I-A and I-B co-express to produce the AB blood type. Multiple alleles. Occurrence of 3 or more alleles controlling the same gene across a population. Human ABO blood types are governed by 3 alleles, I-A, I-B, and I, giving rise to 6 genotypes and 4 phenotypes. Pleiotropy. A single gene influences multiple, seemingly unrelated phenotypic traits by affecting underlying metabolic pathways. For instance, phenylketonuria, PKU, is caused by a single gene mutation that leads to mental retardation as well as reduced hair and skin pigmentation. Polygenic inheritance. Trait variation controlled by 3 or more genes with cumulative additive effects, often impacted by environmental factors. Examples include human skin color, influenced by genes A, B, C, and human height. 3. Chromosomal theory, linkage and recombination, rediscovery, 1900. Hugo de Vries, Karl Correns, and Erich von Schermach independently rediscovered Mendel's principles. Chromosomal theory of inheritance, formulated by Walter Sutton and Theodore Boveri, 1902, who observed that the behavior of chromosomes during cell division mirrors the segregation patterns of Mendelian genes. Experimental proof. Thomas Hunt Morgan confirmed the theory using fruit flies, Drosophila melanogaster, due to their short 2-week life cycle, high reproduction rate, clear sexual dimorphism, and distinct hereditary traits. Linkage. The physical association of genes on the same chromosome that prevents them from assorting independently. Recombination. The process by which non-parental allele combinations are generated through meiosis. Genetic mapping. Alfred Sturtevant used recombination frequencies between linked genes to measure physical distances and create gene maps, a foundation for modern genome sequencing. 4. Mechanisms of sex determination. Types of systems. Male heterogamety. Males produce 2 distinct types of gametes. OXY system. Females are XX and males are XY, e.g., humans and Drosophila. OXO system. Females are XX and males have a single X chromosome designated as XO, e.g., grasshoppers. Female heterogamety, ZW system. Females produce 2 distinct types of gametes, ZW, while males carry 2 matching sex chromosomes, ZZ, e.g., birds. Haplodiploid system, observed in honeybees. Females are diploid, 2n equals 32, and hatch from fertilized eggs. Males, drones, are haploid, n equals 16, and develop from unfertilized eggs via parthenogenesis. Because drones produce sperm through mitosis, they lack fathers and sons, but have grandfathers and grandsons. Sex determination in humans. Humans possess 22 pairs of autosomes and 1 pair of sex chromosomes. Females carry 2 X chromosomes, XX, while males carry 1 X and 1 Y chromosome, XY. Every pregnancy carries an equal 50% probability of conceiving a male or female, dictated solely by whether an X or Y bearing sperm fertilizes the egg. 5. Mutations and pedigree analysis. Mutation. An alteration in the DNA sequence that leads to changes in genotype and phenotype. Mutations can be point mutations, alteration of a single base pair, or frame shift mutations, insertions or deletions of bases. Mutagens, such as UV radiation, induce these DNA changes. Pedigree analysis. A tree diagram mapping inheritance patterns of specific traits or genetic conditions across generations of a family line. 6. Human genetic disorders. A. Mendelian disorders, single gene defects, color blindness. An X-linked recessive disorder impairing red or green cone photoreceptors. Affects roughly 8% of males and 0.4% of females. Hemophilia. An X-linked recessive condition that impairs essential blood clotting factors, causing prolonged bleeding from minor injuries. Sickle cell anemia. An autosomal recessive condition caused by a point mutation swapping glutamic acid, GLU, for valine, VAL, at the 6th position of the beta-globin chain, GAG to GUG. Red blood cells deform into a sickle shape under low oxygen levels. Phenylketonuria, PKU, an autosomal recessive metabolic disorder caused by the absence of the enzyme phenylalanine hydroxylase, resulting in phenylalanine buildup and severe mental retardation. Thalassemia. an autosomal recessive quantitative blood disorder marked by reduced synthesis of alpha or beta globin chains, leading to severe anemia. B. Chromosomal disorders, abnormal chromosome counts, caused by non-disjunction during cell division, resulting in the addition, trisomy, or loss, monosomy, of whole chromosomes. Down's syndrome, trisomy 21, caused by an extra copy of chromosome 21, total of 47 chromosomes. Traits include short stature, a small rounded head, furrowed tongue, broad palm crease, and physical, mental developmental delays. Klinefelter syndrome, 47, XE, caused by an extra X chromosome in males. Traits include an overall masculine body with development of feminine characteristics, e.g., gynecomastia, individuals are sterile. Turner's syndrome, 45, XO, caused by the absence of one X chromosome in females. Traits include sterility due to underdeveloped, rudimentary ovaries, and a lack of secondary sexual characteristics. Chapter 6. Detailed summary of attached file. In text only. Avoid charts. Summary of Chapter 6. Evolution 1. Origin of life, cosmic context. The universe is approximately 13.8 billion years old, formed through a massive explosion described by the Big Bang theory. Earth formed about 4.5 billion years ago within the solar system. Early Earth atmosphere. Early Earth lacked an atmosphere. The surface was covered by water vapor, methane, carbon dioxide, and ammonia released from molten mass. Solar UV rays broke water into hydrogen and oxygen. Escaping hydrogen allowed oxygen to combine with methane and ammonia to form water and ozone. Appearance of life. Life originated approximately 4 billion years ago, 500 million years after Earth's formation. Theories on the origin of life. 1. Panspermia. The belief held by some early thinkers that life spores were transferred to Earth from outer space. 2. Spontaneous generation. The idea that life arose spontaneously from non-living, rotting matter. Disproved by Louis Pasteur's sterilized flask experiments. 3. Chemical evolution. Apparent and Haldane proposed that life arose from pre-existing non-living organic molecules formed under reducing conditions, high temperatures, lightning, methane, and ammonia. 4. Miller's experiment, 1953. S. L. Miller replicated early Earth conditions in a lab flask using electric discharge with CH_4, H_2, NH_3, and water vapor at 800 degrees Celsius, successfully synthesizing amino acids. Early life forms. Non-cellular giant macromolecules, RNA, proteins, originated approximately 3 billion years ago, while the first single-celled cellular organisms arose in aquatic environments approximately 2 billion years ago. 2. Theories of evolution, special creation theory. Conventional belief that species were created intact, diversity remains unchanged, and Earth is 4,000 years old, strongly challenged during the 19th century. Darwinian natural selection. Based on observations during his voyage on HMS Beagle, Charles Darwin concluded that existing life forms share similarities with ancient extinct organisms. Evolution occurs through natural selection, driven by reproductive fitness. Alfred Wallace. Naturalist who independently arrived at conclusions similar to Darwin's while working in the Malay archipelago. 3. Evidence for evolution, paleontological evidence. Fossilized remains found in successive sedimentary rock layers show that life forms changed across geological time spans. Embryological evidence. Ernst Haeckel proposed common embryonic traits among vertebrates, e.g., vestigial gill slits. But Carl Ernst von Baer disproved this by showing that embryos do not pass through adult stages of other species. Comparative anatomy. 1. Homologous structures. Features sharing similar anatomical origins but serving different functions due to divergent evolution, e.g., forelimb bones in humans, cheetahs, bats, and whales. Plant thorns and tendrils in Bougainvillea and Cucurbita. 2. Analogous structures. Features with different structural origins performing similar functions due to convergent evolution in similar habitats, e.g., bird versus butterfly wings, octopus versus mammal eyes, sweet potato roots versus potato stems. Biochemical evidence. Similarities in proteins, genes, and metabolic pathways among diverse organisms point to common ancestry. Anthropogenic selection. 1. Industrial melanism. In pre-industrial England, 1850s, white-winged moths were predominant on lichen-covered trees. Post-industrial soot, 1920s, darkened trees, causing dark melanized moths to survive better against predators. 2. Rapid resistance. Fast evolution of pesticide-resistant insects and antibiotic-resistant microbes demonstrates natural selection operating on human timescales. 4. Adaptive radiation. Definition. The evolutionary process where different species originate from a common ancestor in a specific area and radiate into diverse ecological niches. Key examples. 1. Darwin's finches. Diversification of beak structures on the Galapagos Islands from seed-eating ancestors into insectivorous and vegetarian forms. 2. Australian marsupials. Evolution of diverse marsupial species from a single ancestral stock within Australia. Convergent evolution. Occurs when isolated geography supports multiple adaptive radiations, causing placental mammals to physically resemble corresponding marsupials, e.g., placental wolf versus Tasmanian wolf. 5. Mechanisms of evolution, Darwinian view. Evolutionary change is gradual, continuous, and directional, driven by natural selection and branching descent. Lifespans dictate evolutionary speeds, bacteria adapt in days. Vertebrates require millions of years. Lamarckism. Disproved hypothesis claiming evolution occurs through the inheritance of acquired traits via use and disuse of organs, e.g., neck elongation in giraffes. Mutation theory. Hugo de Vries proposed that evolution occurs via saltation, large, sudden, random, and directionless single-step mutations, rather than minor Darwinian variations. 6. Hardy-Weinberg principle, genetic equilibrium. Gene and allele frequencies in a stable population remain constant across generations unless acted upon by external forces. Expressed as p squared plus 2pq plus q squared equals 1. Disruptive factors. 1. Gene migration, gene flow. 2. Genetic drift. 3. Mutation. 4. Genetic recombination. 5. Natural selection, founder effect. Extreme genetic drift where a migrating subgroup establishes an isolated population with distinct allele frequencies. Modes of natural selection. A. Stabilizing. Favoring individuals with mean trait values. B. Directional. Shifting population traits toward one extreme. Circa disruptive. Favoring extreme phenotypes on both ends of the distribution curve. 7. Geological timeline in human evolution. 1. Evolutionary timeline. A. 2000 Maya. Emergence of first cellular life forms. B. 500 Maya. Invertebrates appeared. Circa 350 Maya. Jawless fish and lobe fins, e.g., coelacanth, evolved, giving rise to amphibians. Plants were the first to colonize land. D. 200 Maya. Reptiles dominated. Aquatic reptiles, ichthyosaurs, returned to water. E. 65 Maya. Dinosaurs, such as Tyrannosaurus rex, became extinct. F. Mammalian lineage. Shrew-like viviparous mammals emerged. Continental drift protected Australian marsupials from placental competition. 2. Human evolution sequence. A. 15 Maya. Primates Dryopithecus, ape-like, and Ramapithecus, man-like. B. 3 to 4 Maya. Upright walking hominids in East Africa. Circa 2 Maya. Australopithecines lived in grasslands, used stone tools, and ate fruit. D. Homo habilis. First hominid human-like species, brain capacity 650 to 800 cc, non-meat eater. E. Homo erectus. Emerged approximately 1.5 Maya, brain capacity approximately 900 cc, ate meat. F. Neanderthal man. Lived 100,000 to 40,000 years ago, brain capacity 1,400 cc. Used hides and buried dead. G. Homo sapiens. Originated in Africa during the Ice Age, 75,000 to 10,000 years ago. Prehistoric cave art developed approximately 18,000 years ago, e.g., Bambetka, followed by agriculture and modern settlements approximately 10,000 years ago. Chapter 7. Detailed summary. Biology in Human Welfare and Human Health and Disease Profile. M.S. Swaminathan, 1925. Background. Born in August 1925 in Kumbakonam, Tamil Nadu, M.S. Swaminathan completed his graduation and post-graduation in botany at Madras University. Research and achievements. He established expertise in genetics and plant breeding, founding the School of Cytogenetics and Radiation Research at the Indian Agricultural Research Institute, IARI. Agricultural contributions. Developed short-duration, high-yielding varieties of rice, including scented basmati, and introduced concepts such as crop cafeteria, crop scheduling, and genetic improvements in yield and quality. Green Revolution and programs. Collaborated with Norman Borlaug to introduce Mexican wheat varieties to India, sparking the Green Revolution. He also initiated the Lab to Land movement and several food security and environmental programs, earning the Padma Bhushan alongside numerous awards. Overview of Unit 8. Biology and Human Welfare, Historical Context. Biology is a younger, formalized discipline compared to physics and chemistry, but the 20th and 21st centuries highlighted its direct utility in human welfare, health, agriculture, food processing, and diagnostics. Key scope. The unit explores how biological advancements, such as antibiotics, synthetic drugs, anesthetics, and high-yield crops, have increased human life expectancy and transformed social structures. Chapter 7. Human Health and Disease. 1. Concept of health. 1. Historical view. Early Greek, Hippocrates, and Indian Ayurveda systems viewed health as a balance of bodily humors. The good humor hypothesis suggested that persons with black bile had a hot personality and fevers. 2. Disproof. William Harvey disproved the good humor hypothesis through his discovery of blood circulation and by measuring body temperature with a thermometer. 3. Modern definition. Health is defined as a state of complete physical, mental, and social well-being, not merely the absence of disease or physical fitness. 4. Factors influencing health. A. Genetic disorders inherited from parents. B. Infections caused by foreign agents. Circa lifestyle choices, including diet, water, rest, exercise, and habits. 5. Disease categories. A. Infectious diseases. Transmitted easily from one person to another, e.g., common cold, AIDS. B. Non-infectious diseases. Non-transmissible conditions, e.g., cancer. 2. Common infectious diseases in humans. Bacterial diseases, typhoid, caused by Salmonella typhi. Enters the body through contaminated food and water, migrating to the small intestine and blood. Symptoms include sustained high fever, 39-carat CIRCC to 40-carat CIRCC, stomach pain, weakness, headache, and constipation. Diagnosis is confirmed via the Weedle test. Note: Mary Mallon, Typhoid Mary, spread the disease for years as a carrier cook. Pneumonia, caused by Streptococcus pneumoniae and Haemophilus influenzae. Infects lung alveoli, filling them with fluid and causing respiratory distress, fever, chills, and cough. In severe cases, lips and fingernails turn gray to bluish, spread via droplets or shared utensils. Others: dysentery, plague, and diphtheria. Viral diseases, common cold, caused by rhinoviruses. Infects the nose and respiratory passage, not lungs. Symptoms include nasal congestion, discharge, sore throat, cough, and tiredness lasting 3 to 7 days. Spread through droplets or contaminated objects, fomites. Protozoan diseases. 1. Malaria. Caused by Plasmodium species, P. vivax, P. malariae, P. falciparum. P. falciparum causes malignant, potentially fatal malaria. A. Life cycle. Requires 2 hosts, humans and female Anopheles mosquitoes, vector. B. Mechanism. Mosquitoes inject sporozoites into humans to parasites multiply in liver cells to attack and rupture red blood cells, RBCs, releasing a toxin called hemazoin, causing recurring fever and chills. TO gametocytes form in RBCs and are taken up by another mosquito during a blood meal. 2. Amoebiasis, amoebic dysentery, caused by Entamoeba histolytica in the large intestine. Causes abdominal pain, cramps, and stool with excess mucus and blood clots. Houseflies act as mechanical carriers, contaminating food and water with fecal matter. Helminthic diseases, ascariasis, caused by Ascaris, roundworm, an intestinal parasite. Symptoms include internal bleeding, muscular pain, fever, anemia, and intestinal blockage. Eggs excreted in feces contaminate soil, water, and crops. Elephantiasis, filariasis, caused by Wuchereria, W. bancrofti, W. malayi. Causes chronic, slowly developing inflammation of lymphatic vessels, primarily in lower limbs and genital organs. Transmitted by female mosquito vectors. Fungal diseases, ringworm, caused by Microsporum, Trichophyton, and Epidermophyton. Features dry, scaly lesions on skin, nails, and scalp accompanied by intense itching. Thrives in warm, moist skin folds, groin, between toes. Transmitted via soil, clothes, towels, or cones. Prevention and vector control, personal cleanliness, consumption of clean water, food, and public hygiene, waste management, are crucial. Vector-borne diseases, malaria, filariasis, dengue, chikungunya, require elimination of stagnant water, cleaning coolers, introducing larva-eating fish, gambusia, in ponds, spraying insecticides, and using mosquito nets and wire mesh screens. 3. Immunity. Immunity is the overall ability of the host organism to defend itself against pathogens. Innate immunity. Non-specific defense mechanisms present at birth, consisting of 4 barrier types. 1. Physical. Skin and mucous coatings of respiratory, gastrointestinal, and urogenital tracts. 2. Physiological. Stomach acid, saliva, and tears. 3. Cellular. Phagocytic white blood cells, neutrophils, PMNL, monocytes, natural killer cells, and tissue macrophages. 4. Cytokine. Interferons secreted by virus-infected cells to protect non-infected cells. Acquired immunity. Pathogen-specific defense characterized by memory. 1. Primary response. Initial low-intensity response to a pathogen encounter. 2. Secondary, anamnestic, response. High-intensity response upon subsequent exposure due to immunological memory. 3. Lymphocytes. A. B lymphocytes. Produce proteins called antibodies into the blood, humoral immune response. B. T lymphocytes. Help. B cells produce antibodies and mediate cell-mediated immunity, CMI. CMI is responsible for distinguishing self from non-self and drives organ transplant, graft rejection. 4. Antibody structure. Represented as H_2L_2, containing 2 light chains and 2 heavy chains bound together. Major classes include IgA, IgM, IgE, and Ig. Active versus passive immunity. Active immunity. Formed when the host is exposed to living, dead microbes or antigens. Slow to develop but long-lasting. Passive immunity. Formed by direct injection of ready-made antibodies. Examples include IgA in mother's colostrum, placental antibody transferred to a fetus, tetanus antitoxin, and antivenom for snakebites. Vaccination and immunization relies on the memory of B and T cells. Recombinant DNA technology allows mass production of antigenic polypeptides, e.g., hepatitis B vaccine made from yeast. Allergies, autoimmunity, and lymphoid organs. 1. Allergies. Exaggerated immune response to environmental antigens. Allergens like pollen, dust mites, mediated by IgE antibodies. Histamine and serotonin released from mast cells cause symptoms like sneezing, watery eyes, and breathing difficulties. 2. Autoimmunity. Genetic or unknown breakdown causing the immune system to attack cells, e.g., rheumatoid arthritis. 3. Lymphoid organs. A. Primary. Bone marrow and thymus, where immature lymphocytes differentiate. B. Secondary. Spleen, lymph nodes, tonsils, Peyer's patches, appendix, where lymphocytes interact with antigens and proliferate. Circumalt. Mucosa-associated lymphoid tissue, constituting roughly 50% of total lymphoid tissue in the body. 4. Acquired immunodeficiency syndrome, AIDS, causative agent, human immunodeficiency virus, HIV, an enveloped retrovirus containing an RNA genome. Transmission. Unsafe sexual contact, contaminated blood transfusions, shared needles, or mother-to-child via placenta. Does not spread by touch or physical contact. Mechanism. 1. Virus enters macrophages, acting as an HIV factory, where reverse transcriptase converts viral RNA into viral DNA. 2. Viral DNA integrates into host genome to produce viral particles. 3. Progeny viruses attack helper T lymphocytes, T_H, leading to a progressive decline in T_H cell count. 4. The host becomes severely immunocompromised, falling prey to opportunistic infections, e.g., mycobacterium, fungi, viruses, toxoplasma. Diagnosis and control. Diagnosed using ELISA. Managed, uncurably, with antiretroviral drugs. Preventive efforts are spearheaded globally by WHO and in India by NACO, National AIDS Control Organization. 5. Cancer. 1. Characteristics. Loss of normal regulatory mechanisms and contact inhibition, where contact with neighboring cells limits growth, leading to continuous cell division forming tumors. 2. Tumor types. A. Benign. Confined to original location, non-invasive. B. Malignant. Mass of proliferating neoplastic cells that invade normal tissue. Undergoes metastasis, cells slough off into blood to establish distant secondary tumors. 3. Carcinogens. Physical agents, X-rays, gamma rays, UV rays, chemical agents, tobacco smoke, and biological agents. Oncogenic viruses containing viral oncogenes, or activation of cellular proto-oncogenes (CONC). 4. Diagnosis. Biopsy, histopathological tissue studies, blood counts, radiography, CT scans, and MRI. 5. Treatment. Combination of surgery, radiotherapy, chemotherapy, and administration of biological response modifiers such as alpha interferon to boost immune response. 6. Drugs and alcohol abuse. Common abused substances. Opioids. Bind to opioid receptors in CNS and gastrointestinal tract. Includes heroin, smac, diacetylmorphine, extracted from poppy plant Papaver somniferum latex. Acts as a depressant slowing body functions. Cannabinoids. Interact with cannabinoid receptors in the brain. Derived from cannabis sativa inflorescences, producing marijuana, hashish, charas, ganja, mainly affects the cardiovascular system. Coca alkaloid, cocaine. Extracted from Erythroxylum coca. Interferes with dopamine transport. Acts as a CNS stimulant producing euphoria and increased energy. Excessive doses cause hallucinations. Hallucinogens. Derived from plants like Atropa belladonna and datura. Medicinal misuse. Barbiturates, amphetamines, benzodiazepines, and morphine, sedative, painkiller, abused outside prescribed psychiatric, surgical usage. Tobacco, smoking. Contains nicotine, alkaloid stimulating adrenaline release, raising blood pressure and heart rate. Increases risk of lung, oral cancers, bronchitis, emphysema, and raises blood carbon monoxide levels. Addiction, dependence, and control, adolescence risk. The phase between 12 to 18 years is a vulnerable psychological period. Driven by curiosity, peer pressure, academic stress, or media perception. Addiction and dependence. Repeated use increases receptor tolerance levels. Abrupt cessation causes withdrawal syndrome, anxiety, shakiness, nausea, sweating. Impacts. Drop in academic performance, violent, rebellious behavior, isolation, financial distress, organ damage, liver cirrhosis, and risk of needle-transmitted infections like HIV and hepatitis B. Prevention strategies. 1. Avoid undue peer pressure. 2. Education and counseling to accept failures and channel energy into healthy pursuits. 3. Seeking help from parents and trusted peers. 4. Monitoring danger signs. 5. Seeking professional psychological and medical rehabilitation. Chapter 8. Here is a detailed text summary of Chapter 8, Microbes in Human Welfare from the attached document. Introduction to microbes. Microbes constitute major components of biological systems on Earth alongside macroscopic plants and animals. They belong to diverse groups including protozoa, bacteria, fungi, microscopic plant, animal viruses, viroids, and proteinaceous infectious agents called prions. Microbes exist everywhere. In soil, water, air, inside bodies of animals and plants, as well as extreme environments like thermal vents, geysers up to 100 degrees Celsius, deep soil, thick layers of snow, and highly acidic habitats. Many microbes like bacteria and fungi can be cultured on nutritive media to form visible colonies for research and study. While some microbes cause diseases, many play essential roles in human welfare. 1. Microbes in household products. 1. Curd production. Lactic acid bacteria, lab, such as lactobacillus, grow in milk and convert it into curd by producing acids that coagulate and partially digest milk proteins. Lab improves nutritional quality by increasing vitamin B12 and helps check disease-causing microbes in the human stomach. 2. Fermented foods. Dough used for making foods like idli and dosa is fermented by bacteria. The puffed-up appearance of dough is due to carbon dioxide, CO_2, gas production. Bread dough is fermented using baker's yeast, Saccharomyces cerevisiae. 3. Traditional foods and drinks. Toddy is a traditional drink made in southern India by fermenting sap from palm trees. Microbes are also used to ferment fish, soybean, and bamboo shoots. 4. Cheese production. Different varieties of cheese acquire specific textures, flavors, and tastes based on the microbes used. A. Swiss cheese. Characterized by large holes created by large amounts of CO_2 gas produced by the bacterium Propionibacterium sharmani. B. Roquefort cheese. ripened by growing specific fungi on it to impart a unique flavor. 2. Microbes in industrial products. Microbes are grown in large vessels called fermenters to produce commercial products on a large scale. A. Fermented beverages. Brewer's yeast, Saccharomyces cerevisiae, is used to ferment malted cereals and fruit juices to ethanol. Undistilled beverages. Wine and beer are produced without distillation. Distilled beverages. Whiskey, brandy, and rum are produced by distillation of the fermented broth. B. Antibiotics. Chemical substances produced by some microbes that kill or retard the growth of disease-causing microbes. Penicillin discovery. First discovered by Alexander Fleming as a chance discovery while working on Staphylococci bacteria. He observed that a mold, Penicillium notatum, inhibited bacterial growth. Development. Ernest Chain and Howard Florey established its full therapeutic potential, extensively treating wounded soldiers during World War II. Fleming, Chain, and Florey were jointly awarded the Nobel Prize in 1945. Impact. Antibiotics treat deadly diseases such as plague, whooping cough, Kali-Kansi, diphtheria, gal-gotu, and leprosy, Kusht Raj. C. Organic acids, enzymes, and bioactive molecules, organic acids. 1. Citric acid produced by Aspergillus niger, fungus. 2. Acetic acid produced by Acetobacter aceti, bacterium. 3. Butyric acid produced by Clostridium butylicum, bacterium. 4. Lactic acid produced by Lactobacillus, bacterium. Enzymes. 1. Lipases. Used in detergent formulations to remove oily stains from laundry. 2. Pectinases and proteases. Used to clarify commercial bottled fruit juices. 3. Streptokinase. Produced by Streptococcus, genetically modified, and used as a clot buster to dissolve blood clots in patients suffering from myocardial infarction. Bioactive molecules. 1. Cyclosporin A. Produced by the fungus Trichoderma polysporum, used as an immunosuppressive agent in organ transplant patients. 2. Statins. Produced by the yeast Monascus purpureus, used as blood cholesterol-lowering agents by competitively inhibiting the enzyme responsible for cholesterol synthesis. 3. Microbes in sewage treatment. Municipal wastewater, sewage, contains human excreta, organic matter, and pathogenic microbes. Sewage is treated in sewage treatment plants, STPs, using natural heterotrophic microbes in 2 primary stages. Primary treatment, physical process, sequential filtration, removes floating debris. Sedimentation. Removes grit, soil and small pebbles. Settled solids. Form primary sludge, while the liquid supernatant forms the primary effluent passed to secondary treatment. Secondary treatment, biological process, effluent is continuously agitated in large aeration tanks where air is pumped in, encouraging growth of aerobic microbes into flocks, masses of bacteria associated with fungal filaments. Microbes consume organic matter, significantly reducing the biochemical oxygen demand, BOD. BOD measures the rate of oxygen uptake by microbes and indicates organic pollution levels. Higher BOD means higher pollution potential. Once BOD is reduced, the liquid is moved to a settling tank where flocks sediment to form activated sludge. A small portion of activated sludge serves as an inoculum for aeration tanks, while the rest goes to anaerobic sludge digesters. Anaerobic bacteria digest bacteria and fungi, producing a mixture of gases, methane, CH_4, hydrogen sulfide, H_2S, and carbon dioxide, CO_2, which form inflammable biogas. Environmental initiatives. Plans like the Ganga Action Plan and Yamuna Action Plan were launched by the Ministry of Environment and Forests to build STPs and protect major Indian rivers from untreated sewage pollution. 4. Microbes in production of biogas. Biogas is a mixture of gases, predominantly methane, produced by microbial breakdown of organic matter, used for cooking and lighting. Methanogens. Anaerobic bacteria like Methanobacterium grow on cellulosic materials to produce methane, CO_2, and H_2. Found in anaerobic sewage sludge and in the rumen, stomach, of cattle, where they assist in digesting cellulose. Cattle dung, gobar, contains high amounts of methanogens, making it suitable for generating gobar gas in rural areas. Biogas plant structure consists of a concrete tank, 10 to 15 feet deep, for bio-waste, dung slurry, covered by a floating lid that rises as gas generates. Includes outlets for supplying biogas to houses and removing spent slurry for use as fertilizer. Developed in India primarily through efforts of the Indian Agricultural Research Institute, IARI, and Khadi and Village Industries Commission, KVIC. 5. Microbes as biocontrol agents. Biocontrol uses natural ecological predation and balance to control pests, diseases instead of toxic chemical pesticides and insecticides. Ladybirds and dragonflies. Used to eradicate aphids and mosquitoes, respectively. Bacillus thuringiensis, BT, bacterial spores mixed with water are sprayed onto crops. When insect larvae eat the leaves, the toxin releases in their gut and kills caterpillars without harming other non-target insects. Genetically modified crops like Bt cotton possess insect resistance via inserted Bt genes. Trichoderma. Free-living fungi common in root ecosystems that act as effective biocontrol agents against several soil-borne plant pathogens. Baculoviruses, genus Nucleopolyhedrovirus, pathogens attacking insects, arthropods. Excellent for species-specific, narrow-spectrum insecticidal applications without harming non-target organisms, plants, birds, mammals, or fish. Ideal for integrated pest management, IPM, programs. 6. Microbes as biofertilizers. Biofertilizers are living organisms that enrich the nutrient quality of soil, reducing reliance on polluting chemical fertilizers. Nitrogen-fixing bacteria. 1. Symbiotic. Rhizobium forms root nodules in leguminous plants to fix atmospheric nitrogen into organic forms. 2. Free-living. Bacteria like Azospirillum and Azotobacter fix atmospheric nitrogen directly in soil. Mycorrhiza, fungi. 1. Symbiotic relationship between fungi, e.g., genus Glomus, and plant roots. 2. The fungal symbiont absorbs phosphorus from soil for the plant and confers resistance to root-borne pathogens, tolerance to drought, salinity, and overall growth enhancements. Cyanobacteria, blue-green algae. 1. Autotrophic microbes like Anabaena, nostoc, and Oscillatoria fix atmospheric nitrogen in aquatic and terrestrial environments. 2. Act as key biofertilizers in paddy fields while adding organic matter to enhance overall soil fertility. Chapter 9. Summary of Biotechnology. Principles and Processes. Historical Context and Foundations. Anthropocentric Scientific Evolution. Scientific advancements since René Descartes shifted toward anthropocentric models focused on improving human comfort and biological utility. Emergence of recombinant DNA. In 1972, Stanley Cohen and Herbert Boyer constructed the first artificial recombinant DNA molecule by combining an antibiotic resistance gene from Salmonella typhimurium with a bacterial plasmid vector. Pioneering discoveries. Boyer identified restriction enzymes in E. coli that cut DNA at specific points, leaving overhanging, sticky ends. Cohen developed methods for extracting, modifying, and reinserting bacterial plasmids into host cells. Core Principles of Biotechnology. Definition. The European Federation of Biotechnology, EFB, defines biotechnology as the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. 2 core engineering techniques. 1. Genetic engineering. Modifying DNA or RNA to alter the phenotype of a host organism. Unlike traditional breeding, it allows selective transfer of desirable genes without unwanted genetic traits. 2. Bioprocess engineering. Maintaining sterile conditions to facilitate the large-scale culture of specific microorganisms or eukaryotic cells for producing vaccines, enzymes, and therapeutics. Basic steps in genetic modification. 1. Identification of DNA containing desirable genes. 2. Introduction of the identified DNA into a suitable host. 3. Maintenance of the introduced DNA in the host and its transfer to offspring. Key tools of recombinant DNA technology. 1. Restriction enzymes, molecular scissors. Function and types. Enzymes isolated from bacteria that cut DNA at specific sequences. Exonucleases remove nucleotides from DNA ends, whereas endonucleases cut internal strands at specific recognition sites. Discovery and naming. HIND2 was the first characterized restriction endonuclease, recognizing a specific 6-base pair sequence. Enzyme names derive from the bacterial genus, species, strain, and discovery order, e.g. EcoRI from Escherichia coli Ry13. Palindromic recognition. Enzymes inspect DNA strands and cut at specific palindromic sequences reading identical 5' to 3' on both complementary strands. Sticky ends. Staggered cuts leave single-stranded, overhanging ends that form complementary hydrogen bonds, enabling DNA ligase to seal fragments together. 2. Gel electrophoresis, separation mechanism. Negatively charged DNA fragments migrate toward the positive anode across an agarose gel matrix under an electric field. Sieving effect. Smaller DNA fragments migrate faster and farther than larger ones. Visualization and elution. DNA bands are stained with ethidium bromide and visualized under UV light as bright orange bands. The excised and extracted DNA bands are collected via elution. 3. Cloning vectors, essential vector features. 1. Origin of replication, ORI. The sequence where replication starts. Controls the copy number of the linked alien DNA. 2. Selectable markers. Genes, such as ampicillin or tetracycline resistance genes, used to distinguish transformed host cells from non-transformed cells. 3. Cloning sites. Unique restriction sites required for linking foreign DNA. Multiple sites complicate cloning by creating multiple fragments. Selection methods. 1. Antibiotic inactivation. Inserting foreign DNA into an antibiotic gene, e.g., BAMI site of TET-R in PBR 322, inactivates that gene, allowing selection through differential plating. 2. Insertional inactivation. Beta-galactosidase. Recombinants interrupt the beta-galactosidase gene, producing white colonies in the presence of a chromogenic substrate, while non-recombinants produce blue colonies. Vectors for eukaryotes. Modified non-pathogenic vectors include disarmed Agrobacterium tumefaciens, T. plasmid, for plants and disarmed retroviruses for animal cells. 4. Host transformation techniques, competent host preparation. Hydrophilic DNA is forced into bacterial cells treated with divalent cations, such as calcium, followed by a heat shock treatment, ice to 42 carat, CIRCC to ice. Direct transfer methods. O microinjection. Direct microinjection of recombinant DNA into an animal cell nucleus. O biolistics, gene gun. High-velocity bombardment of plant cells using gold or tungsten microparticles coated with DNA. Processes of recombinant DNA technology, isolation of genetic material. 1. Cell walls are digested using specific enzymes. Lysozyme for bacteria, cellulase for plant cells, or chitinase for fungi. 2. RNA is removed by ribonuclease and proteins by protease. 3. Purified DNA is precipitated out using chilled ethanol and collected via spooling. Cutting and ligation. 1. Target DNA and vector DNA are cut using the same restriction endonuclease and joined using DNA ligase. Amplification via PCR, polymerase chain reaction. 1. Target DNA sequences are amplified up to 1 billion-fold in vitro across repeated thermal cycles. 2. 3 key steps: denaturation, strand separation, primer annealing, binding of oligonucleotide primers, and extension, DNA synthesis. 3. Uses a heat-stable DNA polymerase, TOC polymerase, isolated from the bacterium Thermus aquaticus. Host insertion and recombinant protein expression. 1. Recombinant DNA is introduced into competent host cells, which are grown in culture media to express the target recombinant protein. 2. Continuous culture systems maintain cells in their exponential, log, growth phase to maximize yield. Bioreactors. 1. Large capacity vessels, 100 to 1000 liters, designed to optimize physical parameters, temperature, pH, oxygen, nutrients, and foam control, for large-scale biological processing. 2. Common designs include simple stirred tank and sparged stirred tank bioreactors. Downstream processing. 1. Encompasses post-biosynthetic steps including product separation, purification, preservation formulation, clinical trials, and stringent quality control testing. Chapter 10 Detailed Summary. Chapter 10 Biotechnology and its applications. 1. Introduction and core research areas. Biotechnology involves the industrial-scale production of biopharmaceuticals and biologicals using genetically modified microbes, fungi, plants, and animals. Key applications include therapeutics, diagnostics, genetically modified crops for agriculture, processed food, bioremediation, waste treatment, and energy production. The 3 critical research areas of biotechnology are: 1. Providing the best catalyst in the form of an improved organism, usually a microbe or pure enzyme. 2. Creating optimal environmental conditions through engineering for the catalyst to function. 3. Developing downstream processing technologies to purify the protein or organic compound. 2. Biotechnological applications in agriculture, food production options. Increasing yield can be approached via agrochemical agriculture, organic agriculture, or genetically engineered crop-based agriculture. Although the Green Revolution tripled food production, agrochemicals remain prohibitively expensive for farmers in the developing world. Tissue culture and micropropagation. 1. Totipotency. The capacity of any plant cell or explant to regenerate into a whole plant under sterile conditions in a nutrient medium. 2. Nutrient medium requirements. Must supply carbon sources, e.g., sucrose, inorganic salts, vitamins, amino acids, and growth regulators like auxins and cytokinins. 3. Micropropagation and somaclones. Producing thousands of genetically identical plants, somaclones, rapidly through tissue culture, e.g., tomato, banana, apple. 4. Virus-free plants. Apical and axillary meristems remain free of viruses even in infected plants. Culturing them in vitro yields virus-free plants, e.g., sugarcane, potato, banana. 5. Somatic hybridization. Digesting cell walls to isolate naked protoplasts from 2 plant varieties and fusing them creates hybrid protoplasts, somatic hybrids, such as the tomato plus potato. Genetically modified organisms, GMOs. 1. Genetic modification enhances abiotic stress tolerance, cold, drought, salt, heat, reduces reliance on chemical pesticides, decreases post-harvest losses, improves mineral usage efficiency, and boosts nutritional value, e.g., vitamin A, enriched golden rice. Pest-resistant plants. 1. Bt cotton. Utilizes insecticidal proteins from Bacillus thuringiensis. The inactive protoxin ingested by target insects is solubilized by the alkaline pH of their gut, turning into an active toxin that binds to mid-gut epithelial cells, creating pores that cause cell lysis and death. Target pests include lepidopterans, coleopterans, and dipterans. Specific genes include Cry-IAC and Cry-EEOB, cotton bollworms, and Cry-IAB, corn borer. 2. RNA interference, RNA, a natural cellular defense in eukaryotes used to prevent root infestation by the nematode Meloidogyne incognita in tobacco plants. Agrobacterium vectors introduce nematode-specific genes that produce complementary sense and anti-sense RNA. These form double-stranded RNA, dsRNA, that initiates RNA, silencing the nematode-specific mRNA and preventing parasite survival. 3. Biotechnological applications in medicine. Recombinant DNA processes enable mass production of safe, effective human therapeutics without inducing foreign immune responses. About 30 recombinant therapeutics are globally approved, with 12 currently marketed in India. Genetically engineered insulin. 1. Natural human insulin consists of two polypeptide chains, A and B, connected by disulfide bridges and is synthesized as a pro-hormone containing an extra C-peptide. The C-peptide is removed during maturation. 2. In 1983, American company Eli Lilly synthesized separate DNA sequences for chains A and B, expressed them inside E. coli plasmids, extracted them, and joined them via disulfide bonds to produce mature human insulin. Gene therapy. 1. Involves inserting functional genes into cells or tissues to correct a diagnosed genetic defect. 2. First performed in 1990 on a 4-year-old girl with adenosine deaminase, ADA, deficiency. Patient lymphocytes were grown in vitro, transfected with functional ADA cDNA using a retroviral vector, and reinfused into the patient. Periodic infusions are required because lymphocytes are not immortal. Introducing the gene at early embryonic stages offers a permanent cure. Molecular diagnosis. 1. PCR, polymerase chain reaction. Detects low concentrations of pathogens or gene mutations before symptoms appear by amplifying nucleic acids, used for HIV and cancer detection. 2. Probes and autoradiography. Radioactive single-stranded nucleic acid probes hybridize to complementary DNA. Mutated genes fail to hybridize and do not show up on photographic film. 3. ELISA. Detects infections based on antigen-antibody interactions. 4. Transgenic animals. Animals engineered to carry and express foreign genes are transgenic. Over 95% of all existing transgenic animals are mice. Key purposes. 1. Normal physiology and development. Studying gene regulation and complex factors such as insulin-like growth factor. 2. Study of disease. Serving as human disease models for cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's. 3. Biological products. Producing valuable human proteins, such as alpha-1 antitrypsin, used for emphysema. In 1997, transgenic cow Rosie produced human, alpha-lactalbumin-enriched milk, 2.4 grams per liter, offering a nutritionally balanced alternative for human infants. 4. Vaccine safety. Testing vaccine safety, such as the polio vaccine, on transgenic mice before human use. 5. Chemical safety testing. Assessing toxicity using transgenic animals engineered to be extra sensitive to toxic substances. 5. Ethical issues and biopiracy, ethical standards and regulation. Uncontrolled genetic manipulation requires regulatory oversight. The Indian government established the GEAT, Genetic Engineering Approval Committee, to evaluate the safety and validity of GM research and public introductions. Biopiracy refers to the unauthorized exploitation of biological resources and traditional knowledge of developing countries by multinational corporations without fair compensation. Basmati rice case. In 1997, a U.S. company patented basmati rice crosses with semi-dwarf varieties through the U.S. Patent and Trademark Office, claiming ownership over traditional Indian varieties. Legislative defense. Countries are formulating laws to counter unauthorized patenting of traditional herbal medicines, e.g., neem, turmeric. The Indian Parliament passed the Second Amendment of the Indian Patents Bill to address biopiracy and patent terms. Chapter 11 Executive Overview and Context. Ecology serves as a unifying principle in biological sciences, bridging molecular biology, botany, zoology, and microbiology to offer a holistic perspective on living systems. It explores how individual organisms interact as collective groups, such as populations, communities, ecosystems, and the biosphere, with each other and their physical habitats. Furthermore, ecology addresses critical contemporary challenges, including anthropogenic environmental degradation and socio-political ecological concerns. Profile. Professor Ramdeo Misra, 1908–1998, father of ecology in India, academic foundation. Obtained his PhD in ecology in 1937 from Leeds University, UK, under Professor W. H. Pearsall. Institutional impact. Established postgraduate teaching and research in ecology at the Department of Botany, Banaras Hindu University, BHU, Varanasi, supervising over 50 PhD scholars. Research contributions. Pioneered understanding of tropical plant communities, ecological succession, plant population responses to the environment, productivity, and nutrient cycling in tropical forest and grassland ecosystems. Policy legacy. His foundational work led to the establishment of the National Committee for Environmental Planning and Coordination, 1972, which later paved the way for the creation of India's Ministry of Environment and Forests in 1984. Core concepts and population attributes. Ecology primary operates across 4 levels of biological organization: organisms, populations, communities, and biomes. Scientific inquiries in ecology. Ecologists evaluate biological phenomena using 2 distinct types of inquiries. 1. How-type questions. Focus on the physical or physiological mechanisms driving a process, e.g., how birds produce sound via voice box operations. 2. Why-type questions. Focus on the evolutionary significance or functional purpose of a process, e.g., why birds sing to communicate during breeding seasons. Defining a population. A population is defined as a group of individuals of the same species living in a well-defined geographical area, sharing or competing for similar resources, and potentially interbreeding, encompassing both sexual and asexual reproduction. Key population attributes. Unlike individual organisms, populations possess aggregate demographic characteristics. Birth and death rates, expressed as per capita births and per capita deaths within a specified timeframe. Sex ratio. The proportion of males to females within the population. Age distribution and age pyramids. The proportion of individuals across pre-reproductive, reproductive, and post-reproductive age groups. The shape of an age pyramid reflects growth status. O. Expanding. Broad base with a large pre-reproductive group. O. Stable. Uniform distribution across pre-reproductive and reproductive groups. O. Declining. Narrow base with a smaller pre-reproductive group. Population density, N, evaluated via total numerical count, total biomass, percent cover, e.g., evaluating a single large banyan tree versus numerous small weeds, or relative, indirect indices, e.g., fish caught per trap, tiger census using pug marks and fecal pellets. Population dynamics and growth models. Population density in a given habitat fluctuates dynamically based on 4 core processes. Natality, B, number of births added to initial density. Immigration, I, number of individuals entering the habitat. Mortality, D. Number of deaths in the population. Emigration, E. Number of individuals leaving the habitat. Growth models. 1. Exponential growth model, conditions. Unlimited availability of food and space. Differential equation. Integral form. Characteristics. Produces a J-shaped growth curve when population density is plotted against time. 2. Logistic growth model, Verhulst-Pearl, conditions. Finite, limiting resources. Represents a more realistic ecological model. Carrying capacity, K. The maximum population size a given habitat can sustainably support. Differential equation. Characteristics. Displays an initial lag phase, followed by acceleration, deceleration, and an asymptote, forming a sigmoid, S-shaped curve. Evolutionary life history variations. Populations evolved toward maximizing their Darwinian fitness, high R value, in response to abiotic and biotic habitat pressures. Breeding frequency. Some organisms breed once in their lifetime, e.g., Pacific salmonfish, bamboo, whereas others breed multiple times, e.g., birds, mammals. Offspring strategy. Some produce large numbers of small-sized offspring, e.g., oysters, pelagic fishes, whereas others produce a small number of large-sized offspring, e.g., birds, mammals. Interspecific population interactions. Interspecific interactions occur between populations of two distinct species and are classified based on whether they are beneficial, plus, detrimental, or neutral. 0. Detailed interaction dynamics. 1. Predation, ecosystem roles. Transfers energy across trophic levels. Regulates prey population densities to prevent ecosystem instability. And maintains community species diversity by suppressing competitive exclusion among prey. Experimental evidence. Removal of the predator starfish pisaster from American Pacific intertidal zones caused over 10 invertebrate species to become extinct within a year due to unrestrained interspecific competition. Defense mechanisms. Animals. Camouflage, cryptic coloration, and chemical defenses, e.g., monarch butterfly accumulates distasteful chemicals during its caterpillar stage by feeding on toxic milkweed. Plants, herbivora defenses, morphological adaptations like thorns, acacia, cactus, and chemical defenses like cardiac glycosides in Calotropis, as well as defensive secondary metabolites, nicotine, caffeine, quinine, strychnine, opium. 2. Competition. Definition. Occurs when the fitness, R, of one species is significantly lower in the presence of another, regardless of whether resources are strictly limiting or through interference competition. Key principles and observations. 1. Competitive exclusion principle, GAUS. Two species competing for the same limiting resources cannot coexist indefinitely. The inferior competitor is eventually eliminated. E.g., Abingdon tortoise became extinct in Galapagos Islands after goats were introduced. 2. Competitive release. Experimental removal of a dominant species allows a restricted species to expand its distributional range, e.g., Connell's barnacle experiment where Balanus excluded Thomulus. 3. Resource partitioning. Species avoid exclusion and achieve coexistence by adopting different feeding schedules or foraging patterns, e.g., MacArthur's 5 warbler species coexisting on the same tree. 3. Parasitism, adaptations. Specialized features including loss of unnecessary sense organs, loss of digestive systems, presence of suckers, hooks, and high reproductive capacity. Types. 1. Ectoparasites. Live on the external surface of hosts, e.g., lice on humans, ticks on dogs, copepods on marine fish, cuscuta on hedge plants. 2. Endoparasites. Live inside host bodies, liver, kidney, lungs, RBCs, with complex life cycles involving vectors or intermediate hosts, e.g., liver fluke requiring snail and fish hosts. 3. Brood parasitism. Parasitic birds lay eggs in host nests, where eggs have evolved to mimic the host's egg color and size, e.g., cuckoo laying eggs in a crow's nest. 4. Commensalism, key examples. 1. Epiphytic orchids growing on mango tree branches. 2. Barnacles attached to the back of whales. 3. Cattle egrets foraging near grazing cattle that flush out insects from vegetation. 4. Clownfish living among the stinging tentacles of sea anemones for predator protection. 5. Mutualism, key examples. 1. Lichens. Mutualistic association between fungi and photosynthesizing algae or cyanobacteria. 2. Mycorrhizae. Association between fungi, nutrient absorption, and plant roots, carbohydrate supply. 3. Plant-pollinator relationships. One-to-one co-evolutionary tight relationships, such as fig trees pollinated exclusively by specific wasp species that lay eggs in developing fig fruits. 4. Sexual deceit. Mediterranean orchid of frieze mimics female bee coloration and patterns on its petals to trick male bees into pseudocopulation, successfully completing pollination. Chapter 12 Executive Overview and Context Ecology serves as a unifying principle in biological sciences, bridging molecular biology, botany, zoology, and microbiology to offer a holistic perspective on living systems. It explores how individual organisms interact as collective groups, such as populations, communities, ecosystems, and the biosphere, with each other and their physical habitats. Furthermore, ecology addresses critical contemporary challenges, including anthropogenic environmental degradation and socio-political ecological concerns. Profile. Professor Ramdeo Misra, 1908–1998, Father of Ecology in India, Academic Foundation. Obtained his PhD in ecology in 1937 from Leeds University, UK, under Professor W. H. Pearsall. Institutional impact. Established postgraduate teaching and research in ecology at the Department of Botany, Banaras Hindu University, BHU, Varanasi, supervising over 50 PhD scholars. Research contributions. Pioneered understanding of tropical plant communities, ecological succession, plant population responses to the environment, productivity, and nutrient cycling in tropical forest and grassland ecosystems. Policy legacy. His foundational work led to the establishment of the National Committee for Environmental Planning and Coordination, 1972, which later paved the way for the creation of India's Ministry of Environment and Forests in 1984. Core concepts and population attributes. Ecology primary operates across 4 levels of biological organization: organisms, populations, communities, and biomes. Scientific inquiries in ecology. Ecologists evaluate biological phenomena using 2 distinct types of inquiries. 1. How-type questions. Focus on the physical or physiological mechanisms driving a process, e.g., how birds produce sound via voice box operations. 2. Why-type questions. Focus on the evolutionary significance or functional purpose of a process, e.g., why birds sing to communicate during breeding seasons. Defining a population. A population is defined as a group of individuals of the same species living in a well-defined geographical area, sharing or competing for similar resources, and potentially interbreeding, encompassing both sexual and asexual reproduction. Key population attributes. Unlike individual organisms, populations possess aggregate demographic characteristics. Birth and death rates, expressed as per capita births and per capita deaths within a specified timeframe. Sex ratio. The proportion of males to females within the population. Age distribution and age pyramids. The proportion of individuals across pre-reproductive, reproductive, and post-reproductive age groups. The shape of an age pyramid reflects growth status. 1. Expanding. Broad base with a large pre-reproductive group. 2. Stable. Uniform distribution across pre-reproductive and reproductive groups. 3. Declining. Narrow base with a smaller pre-reproductive group. Population density, N, evaluated via total numerical count, total biomass, percent cover, e.g., evaluating a single large banyan tree versus numerous small weeds, or relative, indirect indices, e.g., fish caught per trap, tiger census using pug marks and fecal pellets. Population dynamics and growth models. Population density in a given habitat fluctuates dynamically based on 4 core processes. Natality, B, number of births added to initial density. Immigration, I, number of individuals entering the habitat. Mortality, D, number of deaths in the population. Emigration, E, number of individuals leaving the habitat. Growth models. 1. Exponential growth model, conditions, unlimited availability of food and space. Differential equation, where R represents the intrinsic rate of natural increase. Integral form, n_t equals n_0 e⁻rt, characteristics, produces a J-shaped growth curve when population density is plotted against time. 2. Logistic growth model, Verhulst-Pearl, conditions, finite limiting resources, represents a more realistic ecological model. Carrying capacity, K, the maximum population size a given habitat can sustainably support. Differential equation, Fracedient equals Rn, left, frac Nk, right, characteristics, displays an initial lag phase, followed by acceleration, deceleration, and an asymptote, forming a sigmoid, S-shaped, curve. Evolutionary life history variations. Populations evolved toward maximizing their Darwinian fitness, high R value, in response to abiotic and biotic habitat pressures. Breeding frequency. Some organisms breed once in their lifetime, e.g., Pacific salmonfish, bamboo, whereas others breed multiple times, e.g., birds, mammals. Offspring strategy. Some produce large numbers of small-sized offspring, e.g., oysters, pelagic fishes, whereas others produce a small number of large-sized offspring, e.g., birds, mammals. Interspecific population interactions. Interspecific interactions occur between populations of 2 distinct species and are classified based on whether they are beneficial, plus, detrimental, or neutral. 0. Detailed interaction dynamics. 1. Predation, ecosystem roles. Transfers energy across trophic levels. Regulates prey population densities to prevent ecosystem instability, and maintains community species diversity by suppressing competitive exclusion among prey. Experimental evidence. Removal of the predator starfish pisaster from American Pacific intertidal zones caused over 10 invertebrate species to become extinct within a year due to unrestrained interspecific competition. Defense mechanisms. 1. Animals. Camouflage, cryptic coloration, and chemical defenses, e.g., monarch butterfly accumulates distasteful chemicals during its caterpillar stage by feeding on toxic milkweed. 2. Plants, herbivora defenses, morphological adaptations like thorns, acacia, cactus, and chemical defenses like cardiac glycosides in Calotropis, as well as defensive secondary metabolites, nicotine, caffeine, quinine, strychnine, opium. 2. Competition. Definition. Occurs when the fitness, R, of one species is significantly lower in the presence of another, regardless of whether resources are strictly limiting or through interference competition. Key principles and observations. 1. Competitive exclusion principle, GAUS. 2 species competing for the same limiting resources cannot coexist indefinitely. The inferior competitor is eventually eliminated. E.g., Abingdon tortoise became extinct in Galapagos Islands after goats were introduced. 2. Competitive release. Experimental removal of a dominant species allows a restricted species to expand its distributional range, e.g., Connell's barnacle experiment where Balanus excluded Thomulus. 3. Resource partitioning. Species avoid exclusion and achieve coexistence by adopting different feeding schedules or foraging patterns, e.g. MacArthur's 5 warbler species coexisting on the same tree. 3. Parasitism, adaptations. Specialized features including loss of unnecessary sense organs, loss of digestive systems, presence of suckers, hooks, and high reproductive capacity. Types. 1. Ectoparasites. Live on the external surface of hosts, e.g., lice on humans, ticks on dogs, copepods on marine fish, cuscuta on hedge plants. 2. Endoparasites. Live inside host bodies, liver, kidney, lungs, RBCs, with complex life cycles involving vectors or intermediate hosts, e.g., liver fluke requiring snail and fish hosts. 3. Brood parasitism. Parasitic birds lay eggs in host nests, where eggs have evolved to mimic the host's egg color and size, e.g., cuckoo laying eggs in a crow's nest. 4. Commensalism, key examples. 1. Epiphytic orchids growing on mango tree branches. 2. Barnacles attached to the back of whales. 3. Cattle egrets foraging near grazing cattle that flush out insects from vegetation. 4. Clownfish living among the stinging tentacles of sea anemones for predator protection. 5. Mutualism, key examples. 1. Lichens. Mutualistic association between fungi and photosynthesizing algae or cyanobacteria. 2. Mycorrhizae. Association between fungi, nutrient absorption, and plant roots, carbohydrate supply. 3. Plant-pollinator relationships. One-to-one co-evolutionary tight relationships, such as fig trees pollinated exclusively by specific wasp species that lay eggs in developing fig fruits. 4. Sexual deceit. Mediterranean orchid of frieze mimics female bee coloration and patterns on its petals to trick male bees into pseudocopulation, successfully completing pollination. Chapter 13. Here is a detailed text-based summary of Chapter 13, Biodiversity and Conservation. 1. What is biodiversity? Biodiversity refers to the sum total of diversity existing across all levels of biological organization, from macromolecules inside cells to entire biomes. The term was popularized by sociobiologist Edward Wilson. Levels of biodiversity, genetic diversity. Variation at the genetic level within a single species over its distributional range. Example. Rauwolfia vomitoria in different Himalayan ranges varies in the potency and concentration of the chemical reserpine. Example. India possesses over 50,000 genetically different strains of rice and 1,000 varieties of mango. Species diversity. Diversity at the species level within a region. Example. The Western Ghats have a higher amphibian species diversity than the Eastern Ghats. Ecological diversity. Variety at the ecosystem level. Example. India, with deserts, rainforests, mangroves, coral reefs, wetlands, estuaries, and alpine meadows, has far greater ecosystem diversity than Scandinavian countries like Norway. 2. Species numbers globally and in India, IUCN, 2004, estimates. Over 1.5 million plant and animal species have been described so far. Global total estimates. Extreme estimates range from 20 to 50 million species. However, a conservative and scientifically sound estimate by Robert May places global species diversity at approximately 7 million. Global proportions. Animals represent over 70% of all recorded species. Plants, including algae, fungi, bryophytes, gymnosperms, and angiosperms, comprise no more than 22%. Insects make up over 70% of all animal species. Out of every 10 animals, 7 are insects. Fungi. The total number of fungal species exceeds the combined total of fishes, amphibians, reptiles, and mammals. Prokaryotes Estimates are uncertain because conventional taxonomic methods are unsuitable for identifying microbial species, and many cannot be cultured in laboratory conditions. India's biodiversity. India accounts for only 2.4% of the world's land area but contributes 8.1% to global species diversity, making it one of the world's 12 mega-diversity countries. India has nearly 45,000 plant species and about 90,000 animal species recorded. Applying Robert May's estimate, over 100,000 plant species and 300,000 animal species in India remain undiscovered. 3. Patterns of biodiversity. Latitudinal gradients. Species diversity is unevenly distributed. It generally decreases from the equator toward the poles. Tropics, latitudinal range 23.5 circin to 23.5 circs, harbor more species than temperate or polar regions. Bird species comparison. Colombia, near equator, approximately 1,400 species. India, approximately 1,200 plus species. New York, 41 circin, approximately 105 species. Greenland, 71 circin, approximately 56 species. Amazon rainforest, South America, represents the highest biodiversity on Earth. It holds over 40,000 plant species, 3,000 fish species, 1,300 bird species, 427 mammal species, 427 amphibian species, 378 reptile species, and more than 125,000 invertebrate species. Reasons for high tropical diversity. 1. Long evolutionary time. Tropics remained undisturbed by frequent glaciations, unlike temperate regions, allowing millions of years for speciation. 2. Predictable climate. Tropical environments are less seasonal, relatively constant, and predictable, promoting niche specialization. 3. Solar energy. Abundant solar energy leads to higher productivity, indirectly fostering species richness. 2. Species-area relationship. German naturalist Alexander von Humboldt observed that within a region, species richness increases with increasing explored area, but only up to a limit. On a standard scale, the relationship is a rectangular hyperbola. On a logarithmic scale, the relationship becomes a straight line, given by the linear equation. Value of z(O) generally falls between 0.1 and 0.2 for small to medium regional areas, regardless of taxonomic group or region. O ranges from 0.6 to 1.2 when analyzing very large areas, like entire continents. For frugivorous birds and mammals across tropical forests of different continents, z equals 1.15. 4. Importance of species diversity to ecosystems. Ecosystem stability. Communities with higher species richness tend to be more stable. They display less year-to-year variation in biomass, show higher resilience to disturbances, and resist invasion by alien species. David Tillman's experiments, outdoor plot experiments, showed that plots with more species had less year-to-year variation in total biomass, and increased diversity directly contributed to higher productivity. Rivet-Popper hypothesis, Paul Ehrlich. O analogy. An ecosystem is like an airplane. Species are like the rivets holding its parts together. O removing a few rivets, species extinction, might not endanger flight safety initially. However, losing many rivets over time dangerously weakens the plane. O removing critical rivets on the wings, key species driving major ecosystem functions, poses a direct and severe threat to overall survival compared to losing minor rivets, minor species. 5. Loss of biodiversity, current status. The Earth is undergoing rapid species loss due to human activities, experiencing the sixth mass extinction. Current extinction rates are estimated to be 100 to 1,000 times faster than pre-human times. Recent extinctions. The IUCN Red List, 2004, documents 784 extinctions in the last 500 years, 338 vertebrates, 359 invertebrates, 87 plants. O extinct species examples: dodo, Mauritius; quagga, Africa; thylacine, Australia; Steller's sea cow, Russia; and 3 tiger subspecies: Bali, Javan, Caspian. Threatened species: over 15,500 species worldwide face extinction threats, including 12% of all birds, 23% of mammals, 32% of amphibians, and 31% of gymnosperms. The Evil Quartet: 4 major causes of biodiversity loss. 1. Habitat loss and fragmentation, the leading cause of extinction. Tropical rainforests have dropped from covering 14% of the Earth's land surface to less than 6%. Example: the Amazon rainforest is being cleared for soybean cultivation and beef cattle pastures. Fragmented habitats particularly harm mammals and birds, requiring large territories. 2. Overexploitation, human greed leading to overharvesting. Example: extinctions of Steller's sea cow and passenger pigeon, overharvested commercial marine fishes. 3. Alien species invasions, non-native species introduced intentionally or accidentally, turning invasive and threatening native species. Example: Nile perch introduced into Lake Victoria caused the extinction of over 200 native species of cichlid fish. Invasive weeds: water hyacinth, Acornia, Parthenium, carrotgrass, and lantana. Example: illegal introduction of the African catfish, Clarias gariepinus, for aquaculture threatens native river catfishes. 4. Co-extinctions. When a host species becomes extinct, obligate organisms dependent on it also become extinct, e.g., host fish and its unique parasites, plant pollinator mutualisms. 6. Biodiversity conservation. Why conserve biodiversity? 1. Narrowly utilitarian. Direct economic benefits such as food, firewood, fiber, construction material, industrial goods, and pharmaceuticals. Over 25% of modern drugs are plant-derived. Bioprospecting offers potential future economic gains. 2. Broadly utilitarian. Indirect ecosystem services provided by nature. Example: Amazon rainforest generates approximately 20% of total atmospheric oxygen via photosynthesis. Other services: pollination by bees, birds, and bats, flood and erosion control, aesthetic and cultural pleasures. 3. Ethical. Every species has an intrinsic right to exist, regardless of its economic value to humans. We have a moral duty to preserve biological legacy for future generations. How do we conserve biodiversity? In situ conservation, on-site, protecting species within their natural habitats by conserving the entire ecosystem. Biodiversity hotspots. Regions characterized by exceptionally high species richness and high endemism, species restricted to that region and found nowhere else, alongside high habitat loss. Globally, there are 34 biodiversity hotspots, initially 25, later 9 added. 3 hotspots cover India, Western Ghats and Sri Lanka, Indo-Burma, and Himalaya. Hotspots cover less than 2% of the Earth's land area, but protecting them strictly can reduce mass extinction rates by nearly 30%. Protected areas in India: 14 biosphere reserves, 90 national parks, 448 wildlife sanctuaries, sacred groves. Forest tracts set aside where all wild plants and animals are venerated and protected due to cultural, religious traditions. Examples: Khasi and Jainsha Hills, Meghalaya, Aravalli Hills, Rajasthan, Western Ghats, Karnataka and Maharashtra, and Sarguja, Chanda, and Bastar areas, Madhya Pradesh. 2. Ex situ conservation, off-site. Removing threatened animals and plants from their natural habitat and placing them in controlled environments where they receive special care and protection. Traditional facilities: zoological parks, botanical gardens, and wildlife safari parks. Advanced techniques. Cryopreservation. Storing gametes of threatened species in viable conditions at extremely low temperatures for long periods. Tissue culture propagation. Rapidly propagating plants in laboratory conditions. In vitro fertilization. Fertilizing eggs outside the body. Seed banks. Storing seeds of diverse genetic strains of commercial crops long-term. Global initiatives. The Earth Summit, Rio de Janeiro, 1992. The Convention on Biological Diversity urged all nations to implement measures for biodiversity conservation and sustainable resource use. World Summit on Sustainable Development, Johannesburg, 2002. 190 countries pledged to achieve a significant reduction in the rate of biodiversity loss globally, regionally, and locally. That's all for today's revision. If you found this video helpful, please like, share, and subscribe to support our mission of providing free, quality education to every student, no matter where they live. Don't forget to press the bell icon so you never miss our upcoming Class 12 Biology revision videos, important questions, MCQs, and last-minute exam tips. Thank you for watching. Keep learning, keep revising, and all the very best for your CBSE board exams. See you in the next video.